I see a few students who are still here. And so when I wrote this, this is such a strange thing to try to summarize your life in 5 sentences. But the thing that I thought that was interesting as I was trying to do that was to think about how, even how much what I do now is different than what I imagined it would be when I was a first-year graduate student or a second-year graduate student. And so it's always nice to know that, if you're excited about what you're doing, that's great. And if you're not excited about what you're doing, it doesn't mean that your life is always going to be unexciting. So many of you I've talked with about some of the ideas that I'm going to talk about today. I'm an engineer, and so I think about things from maybe a slightly different perspective than people who have been primarily trained in the biological sciences. And so I'm hoping that I'll reflect some of that in this talk. So here's a quick summary. My goal is to tell 3 small stories in the allotted time, so hopefully if you're bored, you can take about a 20-minute nap, and then you can wake up and the topic will be new, and so that will be good. The first is just talking about how behavior affects the hippocampal circuit. So Kamran and Hiro talked very well yesterday about some really interesting dynamics of both local field potential activity and spiking at the timescales that are very long. So this circadian timescale, so day long. I'm going to talk a little bit more about shorter timescales, so on the timescales of minutes, seconds. Then we'll just talk a little bit about replay. This topic came up yesterday, so I wanted to go into a little bit of what we've done in that, and then sort of the process that we're making towards doing real-time decoding of replay. All right, so because I'm an engineer, it's sort of I'm required whenever I give a talk to talk about what is the utility of my research. And I noticed that you guys have not yet learned to do this, some of the students especially. So it's actually really valuable, because if anybody ever happens to see the video, then, except now they'll see this part where I'm talking about it, but they think, oh wow, this is not only is it really interesting, really exciting, it's actually really important for things that are applicable to my grandmother or to my aunt or to my child. And much of what we do, we may feel like it's very far from that, but the reality is that diseases like depression, anxiety, post-traumatic stress disorders are incredibly widespread, and they are diseases of the brain. And as we understand brain circuits, I think all of us are hoping that our discoveries will lead to potentially cures. In my particular case, I'm actually interested in the idea of applying brain-machine interfaces to these diseases. So maybe today, most of us are not comfortable with the idea of a brain implant in order to treat depression, but I think that this is something that will, for example, be coming. All right, so here's my picture of the kind of experiments that we do. Thank you to the rats who gave themselves to all of the data that I'm going to show you. Tetrode recording hippocampus and behaving animals. So how many people here are not familiar, for example, with patient HM? So everybody knows who patient HM is. So I should cold call one of the students, but I won't. Okay, so we know that the hippocampus is important for memory originally because of these humans who had very, very interesting deficits when their hippocampus were lesioned. Now, we've reduced that to these rodents where we study space, but the reality is that I think all of us who are studying memory at least believe that the things that we're learning in terms of space are widely applicable to these broader questions of knowledge and semantic memory. So one of the goals of sort of the last little bit of this talk is trying to understand how we can analyze data that may not be spatial. So we hope eventually, in my lab, I think, as a field, to move beyond mazes. There may be some people in the world who are convinced that the hippocampus is really only about space, but I think most of us believe that in fact it's also important for knowing who the president is or what what bar makes the best caparino. So this is knowledge, and we would like to be able to study this in an animal model, but we don't at the moment, I think, have good analytical techniques that allow us to do things with memories like that. So Just keep that in mind, and I'll try to revisit it at the end. All right, so let's start with this. This is the hippocampal circuit. I draw mine slightly differently than some of the other people do. The first bit of the talk, I'm going to concentrate on area CA1. So we like area CA1 because this is the layer of principal cells that are the output area of the hippocampus, right? So these excitatory neurons here are projecting information about memory to the rest of the brain. So I haven't drawn the subiculum, but essentially there's two main routes of output from area CA1. So one is to the prefrontal cortex. Rodrigo was talking about this yesterday. And then one is back to the entorhinal cortex, which is the major input to the hippocampus. So much of what we know about, for example, LTP, LTD was discovered in this circuit because it's very nice because the synaptic projections are well understood and there's very little recurrence. So the only strong excitatory recurrence is in area CA3. Nobody has talked about area CA2. I think that maybe not this decade, but next decade, maybe the decade of CA2. So just keep your eyes open. I think this is going to be something that will be of growing interest, but I'm also not going to talk about it today. All right, so what I want to talk about is how these connections to area CA1, so CA1 is integrating information that it's getting from two different sets of neurons, right? So one set is part of this hippocampal circuit that we think is responsible for memory, and one is an associational area that's getting information from all of the sensory perception modalities and combining it and then sending that information directly. So somehow this signal that goes to the brain that tells the brain about the world from the perspective of memory is being assembled from these two pieces. And I'm interested in understanding how behavior is modulating that and behavior at a fine time scale. All right, so we've already reviewed this idea of place cells. If you stick an electrode into area CA1 in a rat, and you allow that rat to run around in a room anywhere in their environment, then what you'll discover is that about 30 to 50% of the neurons will be very active. The other 50 to 70% will basically be almost completely inactive. And the ones that are active will be they will be active only in a very specific location in space. So it's interesting to me as an engineer because this is a sparse code. This means that we have many, many neurons and they are active very selectively in a very specific place. So at any one time, most of the hippocampus is not active, but in any given environment, so in a big room, a large chunk of the hippocampus will be active. So it's very different than, for example, visual cortex or auditory cortex, where you can provide a stimulus that will activate the neurons very reliably, irrespective of what the animal is doing. Okay, so place cells. So then the other thing that's interesting is that when animals pause their behavior when they're in slow wave sleep, we see these bursts. Carmen talked about this a lot. An oscillation in local field potential, and accompanying that oscillation in local field potential, bursts of activity where much of the hippocampus, many of the cells will be active, so a much larger fraction of the neurons. And in fact, I wonder if anybody will admit it. I remember I went to about probably a year's worth of talks where people would say this. They'd say, 30% of the cells in the hippocampus are active. And at some point I thought, how do they know that? Because how do they see that there's other cells if the other cells aren't active and they're recording the action potentials of the cells? So what's interesting is that the way that we can do this in the hippocampus is that we have these bursts, and in these bursts we get wide activity, and so we can spike sort those cells based on their action potential waveforms, and we can see that they're there, but then when we put the rat in the environment, they start running around and they all go quiet again. And so we see this activity that is now the opposite of sparse, right? It's dense, meaning that many cells are active all at once. And when we zoom in, we see that often that activity is not just random. It's ordered in a way that is related to the way that the animal had been behaving in the environment. So for example, if the animal had been running in a tunnel and these three cells represented these three locations in the tunnel, then during sharp wave ripple burst, we might see these same cells replaying that pattern. And I'm going to talk a lot about the extent to which that is or is not true. Okay, I'm going to hold my... So this is my video. I think we never tire videos. Okay, so what you see here is you see something like 5 neurons that have been isolated, and that sound is just being applied every time one of those neurons spikes. And the time of their spikes was shown on the left, and now the place where those neurons was active is plotted on this, overlaid on the video trace. So in this case, this animal was getting food here. These are food wells. And so he started here, and he came back to the middle, and he got some food. And so what you saw at the very end was you saw one of these sharp wave ripples, and this is really sort of replay is not beautiful, not noisy. It's actually ugly. It's 2 spikes out of 5 that are active, and so I think that this gives you a taste for what the data actually looks like. All right. So maybe the classic model of how the hippocampus was assisting memory is this thing that maybe we call the 2-stage model. So a model where information is encoded and then stored after some period of time in distributed networks in the neocortex. This is called consolidation. And typically the way that psychologists would have thought of this is that we have some process of experience. In this case, this is an animal who is learning that there's food in 3 arms of this maze. And then he goes to sleep, and while he's sleeping, something happens. Maybe what happens is that he remembers what he had done, and he replays it. His hippocampus takes its short-term memory trace, and it broadcasts it to the cortex, and the cortex learns about what had happened. And then over time, we see that this memory is formed by watching his behavior. So we pop him down in this maze, and he gets better and better at grabbing those 3 pellets of food as fast as he can because he's very hungry. So this idea, this sort of model that there's behavior where we encode, and then there's a second state where we consolidate, I think of as so I called it a 2-stage model, 2-state model. So for a long time, I think in our field, we conflated these 2 things. So we conflated the idea that there's 2 stages of learning, so rapid learning and slow learning, with the idea that animals had these sort of 2 states of their behavior, one of which was active exploration, and one of which was this thing which was a combination of quiescence during awake, which we call large irregular activity, and sleep, slow wave sleep. And then, of course, REM is crazy. We don't know what REM is. So there historically are maybe 3 legs to this model, right? So one is this behavior, the story that I just described, right? One is that there are patterns of LFP that we see. So for example, we see theta during the encoding stage, and we see sharp wave ripples during the consolidation stage. And one is that there are actually differences in the synapses in the hippocampal circuit during these 2 states. And so this result in particular was discovered in the late 70s and incorporated into many models, and then I think people sort of had forgotten about it. So when I was in the Frank lab, I had started working with some animals that were where we had injected channel rhodopsin into the mossy fibers of the dentate gyrus, and we were just flashing light on these animals and recording what happened. So this is one of these science accidents, right? So we were actually trying to express channel rhodopsin in CA3, and it turns out if you inject CAM kinase promoter or lentivirus into the hippocampus, CA3 doesn't actually express it. It seems like it doesn't infect those cells, but you get this beautiful expression in the mossy fibers. And so what happened when we were recording in area CA1 and we had flashed the light on these mossy fibers is that we would see these large field potential events. And we realized that these were essentially the same thing as a EPSP that you would evoke with an electrical stimulation, but we were evoking them with optical stimulation. And when we were doing this, we were just, the rat was running around in the maze, and we noticed that, we saw this, which is that when the animal was running around, we would see these small EPSPs, and then when they were still, so you can see a sharp wave ripple here, the EPSPs were larger. So we're interested in that, and then we started looking through the literature, and we realized, oh, whoa, this is exactly what everybody has known for the last 30 years, but we didn't realize it. And so okay, that's interesting. But then we looked at the data a little bit more, and we realized that when we looked at the size of the EPSPs, so this is now not optogenetically evoked EPSPs, this is now electrically evoked EPSPs, that they weren't this sort of nice cluster of like a strong synapse and a weak synapse, but instead if we, we'll see how the figures are working. There's some sort of weirdness with the display, but this axis is the log of speed. Hold on. Okay, good. So this axis is the log of speed, and this is the size of a bunch of EPSPs that have been gathered during behavior in a particular sample animal. So we were interested in this because this is not the signature of two states, right? This is the signature of a synapse that has basically a continuum of strengths and that is able to change very rapidly from being very strong to being medium to being weak. So yeah, very briefly, let me talk about the method here. So the projection from area CA3 to area CA1 is called the Schaefer collaterals. And in the hippocampus, this is the only projection that crosses the hemispheres. And so that means that if we stick a stimulating electrode into one hippocampus and recording electrode into the other, that we can be fairly confident that when we see an evoked potential in the left hippocampus here, that is being caused specifically by the Schaefer collaterals being activated. And what I just said is actually a lie. It's one of these sort of summaries that people say. It's actually the mossy cells and the dentate also cross the the fissure. But I think we know nothing at all about that projection. Basically, I think Cameron was telling me something about the mossy cells, but yeah. Do you know what the mossy cells do? No. Oh no, that was Sebastian. But you also don't know what the mossy cells do. Okay, yeah. So anyway, there's this strange population of cells in the hilus of the dentate that are also crossing the fissure. All right, so we average this over a bunch of animals, and this is what we see. So if we look at the size of our EPSP, it's smoothly decreasing with speed. All right, so the story that I'm going to tell basically started from this observation, that in fact, if what you expect to see is you expect to see a theta state and a LIA state where you have a weak synapse and a weak synapse during theta and a strong synapse during large irregular activity. When you observe animals that are moving around in a novel environment, at least, you don't see this. You see this continuum of synapse strengths. So then we started looking at what was actually happening during the behavior. So one idea might be that intermediate range of behavioral things, so here's when the animals are sitting still and eating, and here's when they're running really fast. And so maybe this intermediate range is something that they really aren't, that's only a few moments when they're accelerating or decelerating. It's not actually a part of their behavior. But then we analyzed the statistics of the behavior in animals that we're exploring in a new environment. We find that, in fact, they spend most of their time in this sort of intermediate range of sniffing around and looking around. And then as they become more and more used to whatever that is that they're doing, the distribution approaches flat. But it never actually, I think what we often imagine is that the distribution should look like this, that there should be a big peak here where they're sitting still and a big peak here where they're running really quickly and not very much in between. But in fact, if you look at them, so these are in half-second intervals, you find something where the distribution is actually roughly evenly spread. So this is just to show that these intermediate speeds are things that probably correspond less to the animal locomoting, meaning moving really quickly in one direction, and more like looking around. So the way that we ask that question is that we project their behavior into a line. So we take our maze and we linearize it, so we make it a straight line, and then we project the direction that the animal is moving at any moment into that line. And so there's a high correlation between the linearized position, the linearized movement, and the 2D movement at high speeds and a very low correlation at low speeds, which I think makes sense. Right. So, okay, so it's not that there's two states of behavior that are then now somehow being randomly, because we don't have enough points in the middle, sampled into this continuum. It seems like there's a continuum of behaviors as well as a continuum of synaptic strengths. So this is an example of what this looks like. So here's an animal sitting still, here's an animal sort of moving around, and here's an animal running. Okay, so we thought, let's revisit all of the other things that we know about the hippocampus. So yesterday people were talking about gamma, they were talking about ripples. This is a spectrum that has been calculated for a bunch of different speeds of an animal as they're exploring in a maze. So essentially what we've done is we've split our experiment time up into 5 half-second chunks, and we've calculated the spectrum of the LFP in each of those chunks, and we've averaged them over all of the chunks that were in the same relative speed range. So that means that these are slices. So slices of spectrum, and then now we've combined them across these different speeds. And so what you see if you look here is you see bands that make sense. So this top area is your sharp wave ripples. And there's a lot of ripples when the animals are not moving very much. This intermediate band is what we call, I don't know, fast gamma maybe. I'll talk about this in a second, but there's some theory that this comes from entorhinal cortex. It probably also corresponds to some spiking activity. And then this low frequency band is what we would call slow gamma. And so now what's interesting about these is that these actually correspond to different inputs to area CA1. So there's some evidence based on correlograms that slow gamma is being generated by CA3. This also comes from where the sources in the sinks for it are in stratum radiatum. There's very good evidence that fast gamma is coming from the entorhinal cortex, based on correlations, based on lesions, and also based on its sources in sinks and near the fissure. And then obviously we also know that, as was described yesterday, that ripples arise, we think, in the CA3 network and propagate to CA1. So what that suggests is that when we look at the spectrum, that we can think of this area as maybe being CA3, and this area as being CA1. And so we wanted to look at, oh, this is terrible. Let's see what happens if we... So if we now look at the power of specifically the slow gamma band or the fast gamma band as a function of speed, and we see this very similar signature to what we saw in the strength of the synapse. So specifically that as the speed is increasing, that synapse from CA3 to CA1 is becoming weaker, and we also see less gamma power. And conversely, we don't have any information about the strength of the synapse that's coming from the entorhinal cortex, but we see that at the same time that this is decreasing, the power in this band is increasing. All right, so that's gamma and speed. So one of the other things that we know the hippocampus does, obviously what I mentioned we think is most important for is learning. And when you're asking an animal to learn, a rat to learn, the easiest thing to ask them to learn about is space. And the way that we do that is that we put them into a new place. And what they do when they're in a new place is they walk around, they sniff, sometimes they pee or they poop, and essentially they make that place familiar by exploring it a lot. And it takes probably a few hours for an animal to become familiarized with an environment. And when we look at this spectrum, we see that we go from something where we have this sort of interesting sharp wave ripples during the first exposure, so these are 15-minute exposures to an environment, to an animal that now still has this sort of gamma signature, but now has lost its sharp wave ripple signature. And so we were interested in understanding how this potentially synaptic strength effect of speed was being modulated by novelty. When we look at gamma power, we find that the slope of that line, so the slope of this gamma as a function of speed effect, is becoming weaker as the environment becomes more novel. Obviously, you could see from the spectrum that this was also true of the sharp wave ripples. I'll show you that in a second. we took advantage of the fact that in this data that we were looking at, we actually had a experiment where the animal was plopped into a novel environment, and then the very next session, he was put into a familiar environment. The very next session, he was put into a novel environment. So we could control for some sort of random changes that are happening from one day to another by asking, just from this, 15 minutes that were 15 minutes apart, were there differences in the way that the network was working? And in fact, yes, there were. So the speed modulation of gamma was very, very high in the novel environment, and essentially almost gone in the familiar environment, and similarly with the fast gamma. All right, so now here is the sharp wave ripple power. So now this is not actually power in the ripple band. Now here we've actually detected these events, these burst events, and this is the strength of that event as measured in standard deviations above threshold. So we set a threshold, which is 3 standard deviations above the mean, and then every time we see something that rises above that, then we gather it and then gather statistics about those events. So this is sort of a standard way of detecting ripples, is you filter in this frequency band, calculate the power, set a threshold on the power, and then when that power exceeds some level, then you say, okay, this is a sharp wave ripple, which is a thing now as opposed to a frequency band. And we see that the power of these sharp wave ripples is decreasing as a function of speed, exactly as we would have expected from looking at the spectrum. The slope of that line approaches 0 as the environment becomes more novel, but of course there's also fewer events here. All right, so this is all field potentials. We think of field potentials as being a signature of the synaptic input to the network, right? So we think of them as being caused by large fluctuations that are synchronized in the dendrites, right? So what about the output? So what about the spiking? Here you see one. So we looked at spiking during sharp wave ripples, and we find that the number of cells that is active per ripple, so now we've just detected ripple events, and the number of cells that is active is also decreasing, though this is not as steep of a slope, as a function of the speed that the animal is moving. So what that suggests is that there's smaller ripples, and those ripples also have fewer cells participating in them, which at some level is probably a tautology, because ripples are the exception of LFP events, this idea that we think that they're primarily being driven by synaptic input. Let me just take an aside, right? I'm supposed to be teaching you guys. So this is something that confused me for a really, really long time. Local field potential is this thing that is wiggles, right? So you're recording spikes and then you're recording wiggles. And people who understand, they talk about, they're like, oh, those are synaptic, that has to do with synaptic events as opposed to spiking events. And you're like, well, I don't understand. Why couldn't you just have a bunch of spikes that are really small and those add up to wiggles and that be the local field potential? And it's confusing. And the reality is that for you to see something that's large, meaning large amplitude, millivolts, hundreds of microvolts, compared to your spikes, which are small, what that means is that a bunch of stuff has to be summed. And it's much easier for that bunch of stuff to be summed by a huge number of dendrites being depolarized. And so in a brain region like the hippocampus, where you have a layer of cells and then you have a bunch of dendrites that are all beautifully aligned with each other and with your electrode potentially, if you get some axon that's activating all of these dendrites, it might not make these cells fire at all, but it's going to produce a bunch of current relative to your electrode, and so it's very easy to see. And so that's why in general we think that local field potentials are arising from synaptic input, because we know that in a coordinated way is depolarizing the dendrites of a bunch of cells. Okay, so ripples are the exception, right? Because they're so high frequency that we don't believe that there's going to be coordinated input that would be able to cause that. Maybe there is, especially if it's coming from interneurons. But also now I think there's some really good modeling evidence that says that the 200 hertz oscillation is really being caused by spikes firing at 200 hertz. And then those action potentials have some energy that's at lower frequency, and so if you add up a bunch of spikes, you're going to get something that is a 200 hertz oscillation. So all that is to say that if we have fewer ripples or smaller ripples, we probably expect that means that there's less firing. So this is then more interesting. So now we looked during theta, and we were asking about what is happening to the place cell activity during behavior. Okay, so we know some things. So for example, we know that the firing rate is nominally increasing with speed for place cells. But we specifically wanted to understand if there was some sort of information difference that was happening as a result of speed modulation. And so the thing that I'm going to show you is residual correlation. So this is a very nice tool if you have an average pattern of activity, like a firing rate map, PSTH, and you want to understand how, on a trial-by-trial basis, something might be affecting how that neuron is changing. And it's relevant if you have a population of neurons and you believe that there's something else happening to those population of neurons besides the mean behavior. And so what we do is we take a bunch of small bins and we calculate the expected number of spikes that we think that we should see in that bin, given that we have some firing rate map. And so in this case, we have place cells. And so we believe that the place field tells us the firing rate that the neuron is active, should be active at. And so we look at the number of spikes that we see and the difference between the number of spikes that we've seen and the expected amount. So that's the correlation, the residual. And then we correlate this between cells at the same time. Right, so this is a residual correlation. And so it tells you something about how the population is being modulated by something else. And so in this case, we were looking at speed, right? So here's the correlation of the residuals. This is a cumulative distribution function. And then on the right, we've taken the absolute value of these correlations and plot them as a function of speed. And what we see is that there's very high correlation between the residual activity of the network at low speeds and that decreases at high speeds. And so in our head, this is the thought is that this is reflecting something about how there's coherent input coming from. Within my mic, just go on. We think that this is something about coherent input. Wow, this one's so much louder. That's crazy. Coming from area CA3 that's different from the sensory information that's coming from area CA1. And so as a result, we get this more strongly correlated network activity at lower speeds than at higher speeds. All right. So, and this effect is also modulated by novelty, though, in an interesting way, where familiar is the light gray and novelty is the dark gray. So we see that there's even less correlation at high speeds in a novel environment. So I still don't know how to explain that. There's something interesting going on. Maybe one of the take-home messages from this first section is that not only can we say that behavior, meaning the animal's speed, is a very coarse measure of their behavior, is modulating this trisynaptic circuit and regulating the balance between the entorinocortical input and the CA3 input, but that novelty is having a huge effect on what's going on, which makes sense. We know that. We know that obviously this is a situation where the hippocampus should be engaged, but it's interesting to try to measure it in things that we can see. All right, so this is our model, basically that we're going between information that's coming from our memory, meaning area CA3 and the gyrus, and information that's being strongly driven by our sensory perceptions, meaning the entorhinal cortex. All right, and so the conclusion for this little first part of the talk is that the state of that the network is in, meaning whether or not it's doing internal processing or external processing, is not the same as the behavioral state if you look at a small time scale. And I think that this is relevant. I think this certainly has led me to very carefully always now think about, what is an animal doing when I'm looking at its brain? Because what it's doing may be changing to a very large extent how the brain is processing information. All right, so from there I want to talk a little bit about awake sharp wave ripples, right? So one of the things that I showed you was that novelty and behavior are modulating these sharp wave ripple events a lot. Okay, so the problem then is that this sort of model that we have, which is that the sharp wave ripples are being responsible for consolidation in this sort of two-state way, is probably not true. Okay, so we put an X there. So the question is, what are ripples? What is happening during sharp wave ripples, and how is that useful for the animal? So this is, I think I have a couple of asides in my talk. So one of the challenges as a systems, when you're doing this sort of systems neuroscience, you're recording a lot of electrodes, recording a lot of neurons, is that you do a huge amount of analysis. I was hearing that Hiro has spent the last 3 years producing the data that he presented to you guys yesterday, right? This is a crazy amount of analysis compared to like the molecular biologists who, plug their numbers into Excel and they're like, oh, ANOVA, click, and then they're done, right? So the challenge is that the information that we get is correlational, right? So what we learn is really based on observing animals and observing their brains. And so we really like to get away from correlation. We'd like to get towards causation. So there's obvious ways of doing that, right? So mucimol or lesions. These are causal experiments. They change the brain, and when we see changes as a result of that, we can be very confident that we have caused them. But they are slow. And so the thing that we're interested in are very transient patterns of neural activity like a ripple. We at least up until optogenetics, really didn't have tools for silencing it. Even now, you can't give an animal a drug that will block ripples selectively, right? And so what we need experiments where we're doing things, where we detect what's going on, and based on what's going on in the brain, we start manipulating it. And so I think that this is sort of the exciting trend for the next few years. Okay, so this is Gabriel Giardot's work. So they took this animal in this maze, the 8-arm maze, and they interrupted the sharp wave ripples selectively while they were sleeping following learning. What do I mean by interrupted the ripples? So here's our LFP ripple, here's the power of this ripple, and then here also is multi-unit activity, right? So this is going from this sparse activity to this dense activity. So you can set up a system to detect these oscillations, and then with very low latency, stimulate electrically. So Michael Zugara showed this in the mid-2000s, that if you stimulated the fibers that go between the two hippocampuses, you get a very large population spike. And then following that, you get long-lasting inhibition that we think is caused by this sort of feed-forward mechanism, long after hyperpolarization, something. Neuroscience, yada, yada, yada. What he showed was that with titrating the current, you could extend that silencing to be about 150 milliseconds, which is exactly the same period of time as the ripple event lasts. And they also came up with a very nice control, which is that we want to, when we're saying that we're interested in blocking ripples, what we want to know is that our perturbation of the brain is not the thing that's causing the effect, right? We want to say that it specifically is that we're blocking ripples that's important. And so the control that they came up with was to apply exactly the same amount of stimulation at some period that was something like 300 to 400 milliseconds after the ripple. So now whatever was going to happen has happened during this burst, and then we silence. So we're still silencing the hippocampus for the same amount of time, but no longer selectively doing it for sharp wave ripples. All right, so this is aside number 2. I had a graduate student who I asked to look at ripple detection algorithms, and I just wanted to give you guys a little taste of, as an engineer, how I approach this sort of thing. So when you're trying to detect something, you usually have some parameters for your algorithm that you can tune, and the space that you're going to be able to tune over will describe something that is a combination of false positives, missed events, and then in the case of real-time detection, latency, right? So for example, if you wait until the ripple is completed, you can be very confident that you have detected a ripple, but you have a very long latency. So your false positive rate, your rate of misses will be low, but your latency will be very high. Conversely, if you if you tune your algorithm so it has a very short latency, then you expect that you will miss some things and that you will also have a very high false positive rate. And so this is showing, for example, that if we pick a level of misses that we're comfortable with, then that will correspond to a different amount of latency. And so this graph is a little hard to read. So the right way to understand this is that these are actually extending down. So pick the number 0, for example, and these extend way out over here. So pick a latency of 45, and what that will mean is that for this algorithm that my student came up with, the miss rate is 0.01, and for these power spectral analyses techniques, the miss rate will be 0.02. And similarly, you'll see something with a false positive rate, where the rate of false positives will increase as your latency decreases. All right, aside done. So this is what Girardot et al. Discovered, which is that when they disrupted the ripples, that this animal learned this maze much more slowly. You could say that they failed to learn to do this task over the course of 2 weeks. And this is really interesting because these rats are being given access to this maze for about half an hour a day, and that has something to do with their satiation, right? They eat enough food that they're no longer interested in doing it. And it actually takes them something like 2 weeks to do the task, to learn to do this ideally. And if we disrupt ripples for just a few minutes after their behavior, for something like half an hour or an hour, that halves their ability to learn, right? So what this is saying is that this very select, very, very short bursts of activity in the hippocampus are supporting this, very large animal's behavior. So this is actually completely consistent with this sort of two-stage model of learning, right? So the animal's encoding, they're consolidating when they're asleep. Now we would say that they're consolidating not just when they're asleep, but maybe specifically during these sharp wave ripples, and as a result, their learning is disrupted. So we were interested in applying this to sharp wave ripples that were awake. And so we built the same sort of system. We added some other features like the ability to combine information from multiple channels. I think if you want robust ripple detection, I think combining from maybe 2 to 4 channels is a good idea. This gives you the ability to say, okay, this is not noise because I've seen it on at least 2 channels. And then we also had to add something that would try to rule out this false gamma from triggering our activity. So we added a speed threshold so the animal could be moving but not moving too fast. And then we also needed a task that the animal would be doing while he was learning nominally. So this task is a W maze. It's a spatial alternation. It's like a T maze. So there's food at the end of these 3 arms, and the rat has to learn 2 rules. So one is that if he's in the outside arm, he returns to the middle. And if he's in the inside arm, he goes to the opposite arm from which he came. So if he had gotten food here and then gotten food here, and he goes here, he will be rewarded. But if he had gotten food here and then he goes here and then he comes back to this well, he won't be rewarded. And that, in fact, is what the rats typically. This is sort of the process of learning this, is that they randomly explore. Usually, we start them always in the middle arm, and they'll get some food there. They'll get really excited, right? They'll run around and they'll come back here. They don't get any food. After a while, maybe they'll go back to the middle arm. They get some food there, and then they're like, I got some food back at this first arm. Let me try going back there, and they don't get any food, and so you get something where they're going back and forth, and then typically what will happen is they said, oh, there wasn't any food here. Maybe there'll be food over on the other side. And then, of course, they don't get rewarded, and they go back to the middle, and so on and so forth. So what we found is not visible because my PowerPoint is not scaling properly. I apologize. What we found is that this first rule, they learn just fine. And what's interesting about that is that that's in contrast to animals that have hippocampal lesions that don't successfully learn this first rule. So by the end of a week, almost 100% of the time, when they left the center arm, when they left an outside arm, they would go to the center arm. But this other rule, this alternation rule, that we imagine having more of a memory load, I have to remember where I was previously, they didn't learn, or they didn't learn as fast. So some animals didn't learn, other animals learned more slowly. And so that was very, very interesting because it suggests that these bursts of activity now are specifically supporting a kind of behavior that involves working memory. Now, the problem, of course, with this result is that this is learning over time, right? So it's unclear whether they're having problems learning to alternate or whether they are having problems remembering what they did, and as a result they're having problems learning to alternate. But in either case, it suggests that the circuit is active online while the animals are behaving and allowing them to do this. Okay, cool. Okay, so here's this summary number 2, right? So I showed you that there's some, during behavior, the hippocampus is doing some dynamic stuff. It's interesting, it's not switching between two modes of synapses, but it's sort of gradually moving between these two things. But we still believe that sharp wave ripple events are these distinct things, and we find that if we take away those distinct things, that we disrupt behaviors that depend on working memory. All right. So as I was leaving UCSF, we'd built this system, and we were trying to understand what really were ripples doing. So the way that normally we would present this, normally that I would present this at that time, was to say that in fact it wasn't the ripples that we were disrupting, but it was replay, right? So let's go forward for a second. So you remember this figure. So this is what happens during ripples when we tell this story, right? Is that there's the play cell activity, and then during the ripples, the animal is replaying the memory. So now, it turns out that this is not true. A large fraction of the time when we see ripples, we don't see replay. And when we're doing this ripple disruption, we're disrupting all of those things, right? And so one of the goals now of my lab is to be able to build a system where we would actually now look not at the ripple, but at the information that is being represented during the ripple, and be able to selectively block or perhaps reward information during replay, and understand how the brain is actually using the spikes that are coming during those ripples by causally and selectively perturbing it. Okay, so that's not trivial. Let's talk about the steps to doing that. So we need ripple detection, but we can sort of check that off. Behavioral tracking, real time, we'll put a half check there. You can buy these commercial systems. We've been working on one in the lab that is not done yet. If we're interested in sleep, then real-time processing of sleep, then we would want EMG. That's easy, that's a check. Okay, now we need real-time spike sorting, right? Because we want to identify individual neurons in order to identify what information is being represented in the replay event. This is difficult, and there is no good solution. And in addition to that, we want to be able to understand what the hippocampus is representing by those spikes very quickly, right? When we pop an animal down into a new environment, they're immediately going to start learning about that environment, and they're going to be telling the rest of their brain through replay what's going on. So as those spikes are being generated, we need to be able to understand what they represent, like different places in the maze, and build models that do that on the fly. And so this is sort of where we are in the process of building our system. And then when we put these things together, we'll have replay detection. So I'm actually going to talk a little bit about this process of the model building and the replay detection and some simulated data that we have for that. But I wanted to give you guys some, so in terms of pedagogy, ideas about building something like this. I'd say that one of the things that I've learned is that it's best to do this, to do a process like this by remembering this design for test idea. Basically that you should figure out what is going to be important for your experiment, what specifications are going to be critical for it to work, and then figure out how you're going to test those specifications. And so in our case, we, for example, cared about latency or the reliability of ripple detection, right? So those are our specifications. And then when you build the design, test it against its specifications. And then that way you'll be confident that it's working. And if you've set up your specifications right for each of these things, and you've tested each of these things against those specifications, and you'll be confident that your whole system will work. And I know that it's very easy, because I've done this, to just try to build the whole thing and not really, and have each of the pieces sort of be working, but you're not sure, there's some flaws, and then you put the whole thing together and it doesn't work and you don't know why, right? So it's always very good to just think very carefully, okay, how am I going to test each step of this along the way? I think simulated data is actually super valuable for this, and that's why we've been pursuing that. All right, so so thinking about the specifications, in order to do replay detection, I realized that we needed to know how noisy replay was. So we needed to understand what the noise statistics of the replay that animals were producing was. And so that led us sort of down this rabbit hole of trying to understand what are these patterns of spikes that are being produced during ripples. Right? So this is the canonical idea that this is replay, but the reality is that it's not always. But let's very quickly talk about replay. So I just showed you that sharp wave ripples are enhanced in a novel environment. They are present and important both during sleep and awake states. Now, so we know this has been shown that replay is enhanced during learning in novel environments. We know that replay during sleep, so this is correlational evidence, is correlated with learning. So if you have more replay, you'll learn better. We know that in certain cases, the information that is in awake replay will relate to an animal's future behavior. And we also know that replay is more likely to be detected in awake animals than in sleeping animals. But this is basically now the extent of this is qualitative knowledge about replay, and the quantitation is essentially is missing right now. All right, so this is how we normally would detect replay, right? So we would find potential events, either based on sharp wave ripples or multi-unit activity thresholds, and we would compare the spikes that we see during those events to what happened during behavior. And I'm going to talk about some ways of doing that. And if we see a pattern that is the same, so if the pattern during the ripple is the same as the pattern during behavior, then we'll do some sort of statistical test. We'll ask, okay, is this something that we would just have expected to see by random chance? And if not, then we say that this was a replay event. And here's the number. So depending on who you ask, between 7 and 50% of potential replay events are replay. And this is not a PDF. This doesn't tell us anything about how that distribution is shaped. But it is a little distressing because it suggests that half the time we don't know what the hippocampus is doing. All right. So very quickly, how do we detect? So how do we do this process? Right. So how do we compare the spikes to what happened during behavior? So this is what I call the poor man's approach. So if you don't have a lot of neurons, you can just look at pairs of neurons, and you can form a pairwise correlation. So basically, you'll find at any point in time, you just correlate the activity of all of your pairs of neurons. And then if you separate those neurons by how far their spatially separated place fields are, so that's this axis. So here's the peak of the place fields of these two neurons, for example. This is something like here. Then you can ask about what the cross-correlogram of the spikes look like. And so this is a bunch of cross-correlograms that have been averaged across neurons and then combined by how far apart the place fields of the neurons were. And so what we see is that when the animal's running, we have this beautiful correlation pattern, right? So you see as they're moving forward, the next cell in space will be very highly correlated with the cell of the place field that we're currently in. Does that make sense to everybody? Does this line make sense? So if you're correlating two place cells and an animal is running like this, and I look at the spikes from this cell and the spikes from this cell, then I expect my cross-correlogram will have a peak that is somewhat shifted in time. That people are nodding. Okay, so that's like right here, right? So this is greater than 0, and also in time because the rat is running. Okay, and so it's interesting. So Cameron talked a little bit about theta sequences, so you can actually see those as little jags in this cross-correlogram. So now you can do exactly the same analysis when the animals are not moving. So now they're not running, and you select for ripple events either by multi-unit activity or by a LFP threshold. During behavior, you see this X that corresponds to awake replay, which we see often goes in both a forward direction or a reverse direction. And during sleep, you also see another X, except there's a bias towards a forward direction. So this is something that we know. And so now you can do statistics. So you can ask, is there more energy here than here, for example? That would be evidence that there was, is there more energy here than here? And that would be evidence that there was replay. Okay, so this is the David Reddish likes this. This goes back Kudramodi and McNaughton, and also in the Buzaki lab, they've used this approach to look for replay when they didn't have very many spikes. When you have a lot of spikes, you want to say, I know exactly what the animal's thinking about. I don't just think that I'm not just looking and seeing that he's moving forward. You want to see that he's dreaming about, running down that third arm of the maze that has the food in it. Okay, so the way that we do this is that we use what's called a decoding approach. And in order to decode, you have to encode. So what is the encoding model? The encoding model is an estimate of a place field. Okay. So who here has tried to estimate a place field before? Okay, maybe like 5 of you. Okay, but people are, I know that there's people who aren't raising their hands, and I see you. Okay, good. So this is not actually that hard, but it's moderately non-trivial. So I thought that this was worth mentioning. So how would you think that you would estimate a place field? So you might think, I'm going to do a PSTH. This is just like, there's just trials. So I have the animals running in this maze, and I'm just going to line up the spikes. But then what you find is that animals run at different speeds. And so the amount of time that they spend in any location changes from one trial to another. And so you can't line up the spikes just like a normal PSTH. So you have to be a little bit smarter. And so the way that we do it is that we look at space first, and then we look at time second. And then we divide space by time, and we get a firing rate map. And so that's basically what this is describing. All right. So for decoding, we now have to make some assumptions. And so the assumption that we will make is that neurons are roughly Poisson within their firing fields. So this is Bayes' rule. So this says that if we're interested in the probability of the rat being in a particular position given the vector of neurons that we've observed spiking, that this is proportional to the encoding model, so the probability of the spikes being generated at that position, multiplied by the prior, so the prior probability of all the positions. So typically when we're doing decoding, we assume a uniform prior, so we cross this out. And this is, for Bayes' rule, this would be normalized by the probability of the spikes that we've observed, but they don't change between different positions. So we can now just write this formula. So this is just saying that we'll take the product across all of the neurons that we've observed of the firing field. So this is the Poisson Pdf. So lambda to the k times e to the minus lambda delta t. So there's actually a delta t here also, but it is the same across all of the potential positions as well. So what this will give us is if we have divided our space into a bunch of bins, this will give us a vector that's of size number of bins by 1, and in each position, in each element in that vector, there will be a probability. And so this is what this looks in practice. So here's a replay event, here's the spikes, they've been ordered by where their place fields were. And then here time is by color, so here is the probability of different positions as a function of time. And then we can project, whatever the peaks or something like this onto this track, and we see, okay, he's remembering running along the maze. So when you see something like this, it still is a little bit non-trivial to figure out how to know that that was not created by chance. And so how do we do this? Okay. So this is where sort of the existing techniques start to break down. So we have to make some assumption, again, about what the rat is dreaming about. And what we'll assume is that he's always dreaming about running forward at a constant speed. And so that what that means is that his position as a function of time should be a straight line. And so we'll take our probability distributions and we'll spread them out in space. And so then, now that gives us something that's probability as a function of time. And we'll just fit a line to that. And we'll calculate something, some score, r squared of that line. Okay, so that still doesn't tell us whether that's significant or not. So the next step is to try to bootstrap significance by generating a bunch of other data and doing that same analysis on them, and then looking at the R-squareds or whatever the score is of the lines that we have generated with our artificial data. And so here are the typical shuffles that people do. So one is to say, okay, I'm going to take my vector of estimates, that's a vector of probability as a function of time, and I'm going to randomly shuffle them. So I'm just going to reselect which position goes with which time. And then I'll fit that line, and then I'll calculate, I'll do that 1,000 times. That will give me some distribution of scores. And then I'll say, okay, what's the tail over here, and where's this R squared of my actual event? And you hope that it's over here, it's bigger. And then you say, okay, this is significant. So that controls for the idea that the animal must not, our model of replay is that the animal does not teleport randomly around the world. Who knows whether or not that's a good model. And that controls for that, right? So what we're doing is we're taking the same replay, but we're turning it into random teleportations. And then the other thing that people typically do is to randomly shuffle which neuron is being assigned to which spike. And so this controls for one or two neurons that are bursting, and as a result, you've seen two spikes from two neurons, and you say, okay, well, this must have been a replay event. But now if you replace them with other neurons, then the R squared is exactly the same, because for any two neurons that had that pattern, you would have gotten a line. All right, and then a less common control is to just sample from other events. So now we'll say, okay, I want to make sure that for a potential replay event of this size, this number of time points, that it wouldn't just always be considered significant. All right, and so then when we do that, as I said, we get a number that's like 50%. Okay, so let's ask this question. What is happening during ripples? So what I've just described is that a lot of the time there's replay. So what else could be happening? I don't know what is there anything that I haven't thought of? So one is that the animal's actually thinking about another environment. So let's say that we're recording our animal in his home cage, and we don't see replay of the home cage, we see replay of something else. And it's probably because he did something really exciting today. We took him out of the vivarium, we took him up, we put him in this maze, we gave him chocolate sprinkles. It was his first time to eat chocolate sprinkles. They were so good. Of course, that's what he's remembering, right? And so we shouldn't be surprised if we don't decode that as replay of the home cage, because he did something else that was much more interesting that he should be replaying. Okay, so there's one experiment that I know of where they've put an animal in two mazes, and they've looked at the replay, and then basically the amount of replay that they detect rises from 50% to 55% if they include the two environments. So it seems like that doesn't explain everything. That doesn't explain everything. The second bullet point is that our algorithm for detecting replay could be bad. We're just excluding things that really are replay. And then the fourth possibility, which is either the most exciting or the most depressing, depending on how you think about it, is that the hippocampus is just producing noise. And it's always producing this random noise, and maybe the random noise is really interesting for something. Maybe it is actually what allows our imagination to work, right? So maybe the rat starts with this, he's dreaming about the maze, and then he starts thinking about, oh, wouldn't it be cool if there was 4 arms to the maze? There'd be more milk, it would be so great. And then in some experiments, so for example, there's this classic experiment where the animal is running in a figure 8 pattern. So you can imagine he's running like this. This is not a good example. Running in a figure 8 pattern, and they see events where he imagines running across the top of the maze, which he's not allowed to do. So he's imagining a behavior that he's not allowed to do, but actually is a shortcut that would get him the food much faster, right? So this is exactly the kind of mechanism that we might imagine would underlie creativity, right? So the ability to take what we've learned and sort of manipulate it into something new that's even better than what we've learned. But what are the statistics? And how can we even begin to quantify whether something, for example, is similar to replay of an environment, but slightly different? Okay, so in the last 3 minutes, I'm just going to talk about what we've been doing towards developing this. So we've been trying to treat replay as a So this idea of the thing that you're remembering as a latent variable. So latent variable basically means something that's not observed. And so this is a sequential latent variable model. So this is a hidden Markov model because we assume that when you're transitioning from these latent states, that your probability of transmission only depends on the state that you're currently in. And then every state corresponds to some observed neural activity. And so in this case, our observed neural activity is going to be firing rates that we assume are probably something like place fields. And so what's nice about these models is that you can just take spiking data, so you can take observations, and then you can fit them. And it's non-trivial and takes a lot of data. So this is how we would use this sort of model to ask whether a replay event that we're seeing is good or bad, or is close or far away. So this is our state transition matrix. So this is showing the probability of transitioning from state 1 to state 2, from state 2 to state 3 is in this box, from state 1 to state 3 is in this box. Okay, so based on the structure of this maze and our, in this cartoon example, I know exactly what these states are. So I know that the animal should go 1, 2, 3, 4, 5, 6, 7, or 1, 2, 3, 4, 8, 9, 10, right? So that corresponds to these elements in the transition matrix being non-zero. So what we see is two possible behaviors, right? So one is that he can go like this, and one is that he can go like this. Does that make sense? So these vectors correspond to us observing a sequence of states. So we've observed state 1, then now once we're in state 1, we know that we must go to state 2. The next thing we observe is state 2, we observe state 3, and then we either observe 5, 6, 7, or 8, 9, 10. So something like this is highly unlikely. And so if we observe a sequence of states that looks like 6, then 1, then 6, then 3, then 4, then 3, this is not likely, because this corresponds to this animal doing something like this, right, which he physically can't do, and we assume that his brain would not want to produce that pattern. All right, so in the absence of testing this on real data yet, which hopefully we'll be doing in the next few weeks, we generated some synthetic data. Now we know exactly what has happened. We know exactly what the firing rate patterns of the neurons are. We know exactly what the animal is doing. And in fact, to make things easier, we're just going to have the animal be running in a circle. So this is the classic Kemere maze, right? So the animal is running in a circle on a maze. And then we're going to assign circular Gaussian, so these are von Mises distributions, so circular Gaussian place fields. We'll just pick 100 neurons, and because we have the statistic that in a novel environment 50% of the neurons are active, we will assign them 50 of the neurons to have random place fields along this maze. Okay, so we're actually going to do 5 of these mazes to represent 5 different environments that we could potentially be producing replay from. And then we're going to add some noise that I'll talk about when we get to that. All right, so we've now calculated the probability of a replay event that we've generated from that environment. So we've generated now something like 10,000 replay events. And we've generated them from all 5 of these mazes. And now what we're asking is, for maze number 1, when we see a replay event, what is the likelihood that that replay event came from maze 1, and what is the likelihood that it came what is the likelihood that it came from maze 1? So maze 1 corresponds to this black maze, and what we see is that the likelihood is some number. So likelihoods just sort of scale. They become very negative. This is actually the log likelihood. So this is a very large, small number, a very small number that scales with the number of neurons and all these sorts of things. So the likelihood of the maze replay events, given that we think that we're observing maze replay events, is here. It's very narrow, and it's close to, it's large. So the probability of replay events generated from the other environments is over here. So it's much, much smaller, and it's also much wider. And so this is cool, because this says that if we observe something like this in our actual replay data, if we observe one mode of replay scores here, and then a second mode of replay scores here, that our hypothesis would be that this animal is really, it's generating multiple patterns, but those patterns are corresponding to different environments, right? And we know one environment, we don't know the other environment. But that's not the only thing we could observe. So we added some other random ideas of what the animal could do. So this dotted line is the rat now, instead of running in a circle, he runs for a little while in one direction, he turns around, he runs back, and then he runs forward again. And so what we see is that when we look at the replay of that sort of behavior, that it produces a score that is smaller, but it's not that much smaller than what we would expect given the animal's, the model behavior that we believe, which is the animal is running in a circle. So now if we allow him to teleport, so now he's hopping between different locations, which he can't do physically, obviously, then we see that now we get a score that is probably distinguishable from this first mode, but is still overlapping a little bit. And then finally, now if we try to just generate something that's very noisy, so this is not informative at all, this is just us generating random sequences of spikes, we see that we get this very broad distribution that is clearly separable from what we would expect to see. So what this says is that if what we observe is a sort of a broad, one broad hump and then a second sort of mode, what we would now suggest, what we now expect that means is that the brain is producing replay of the environment that we've observed, and maybe not producing replay of other environments, but actually producing sort of noise, right? So by looking at the width of these distributions and their relative heights, we can say something about what actually is happening with replay, and then that in turn will allow us to build these real-time systems that we want to build, and then everybody will be happy. And so that is the conclusion. Everybody will be happy. So the first part of the talk I did as a postdoc with a graduate student named Maggie, who is now working for a consulting company, and Shantanu Jadhav, who just started a job at Brandeis University, and Lauren Fank, obviously. And Etienne Ackerman is the graduate student who's working on these latent variable models of replay. And I thank you for your attention.